So ERESA is an EGFR inhibitor, a small molecule, and it's well known that the EGFR pathway is linked to a variety of intracellular events which impact on tumour development and proliferation. Now, the first ERESA was developed on the basis that tumour cells showed EGFR protein, an enhanced EGFR protein, so it was assumed that that's what it would be targeted at. And phase 2 studies did indeed show that there was some anti-tumour activity. But then what happened was that a variety of pieces of work happened during phase 2, which showed that it wasn't actually working the way it was expected to work. So we worked with Dako cytomation on the identification of some factors that influence clinical response. And there was some evidence that gene expression of EGFR did predict sensitivity, but it didn't really hold up. So there were various other studies, and proteomic studies, and epigenomic studies, and then the important piece of information was that inherited variants of EGFR and survival might be linked. And then we did some more studies, and then the important thing was that gene mutations were identified that made lung cancer respond to ERESA. So that a huge amount of potential biomarkers were tested out. Copy number was one that was felt to be really important as well. And this was all while the clinical trials were going on. So because the clinical trials had not been expected this type of result, they were carried out in the larger population. So the biomarkers all had to be evaluated retrospectively. So in this particular case, we can see that the data has been split into mutation positive and mutation negative individuals. Now, you can see these are Kaplan-Meier curves, and I think in the mutation negative you can see that the placebo is the blue and the gefitinib is the green. There's very little difference between the two curves, whereas the mutation positive, the proportion surviving is much better with ERESA. And that was similar result was for copy number, mutation, expression, so it was still not clear actually what was driving this. And none of these results that we got were actually clinically, statistically significant. And that's the trouble with retrospective data, it generally doesn't give you that information. I'll go back a bit later to say where ERESA is now, but it just shows you an example of what happens when you come in a bit late with trying to identify and type your markers. This is an example of, I'd call it a failure, maybe I should call it a challenge really. It's Exanta. It didn't fail because of personalised healthcare, but it could have been saved because of personalised healthcare. So Exanta was a thrombin inhibitor. And it was in its phase 3 trial, so it had been quite an expensive operation. And there were some safety concerns because there were raised liver enzyme levels in some patients. Didn't get to clinical significance, but it was certainly of concern. And when we looked at all the phase 3 data, the majority of the patients with these ALAT elevations were in the north of Europe, which gave you a suspicion that it might be a genetically determined effect. So we were already in phase 3, and I should say, because I was in the genetics department at the time, we had been trying to persuade all our clinical development teams to start collecting samples a long way back, but Exanta was one of those that hadn't. So we had to retrospectively go back and collect all the cases where there had been raised liver enzymes and some controls as well. And we undertook a genome-wide scan in collaboration with a biotech group in the US. And I have to say that, do a genome-wide scan but discover a biomarker isn't as easy as all that, because we had to set up quite a lot of data analysis, etc. But we did discover a biomarker, and the biomarker we discovered was the HLA region. So it was obviously an immune response that was happening with these patients. It in itself was not a sensitive enough marker for clinical use. I think it predicted about maybe 70%. But we couldn't go any further because Alexander happened to be withdrawn at about that same time, because there was one serious liver toxicity. So it was actually withdrawn. Now, if we had started looking at potential safety marker at a very early stage, and it probably did show up at a very early stage, then it's quite possible that that drug might be on the market today. Okay, to go back to the drug diagnostic and co-development, I guess the point I'm trying to make is you have to be really early with your biomarker to make personalised healthcare work. So you need to, and I think in the case of AstraZeneca, we're doing a huge amount around trying to get those biomarkers discovered early. And we don't have diagnostics groups within AstraZeneca. We used to, some people might remember. We divested ourselves, which is probably a sure sign that diagnostics was about to become really important. We divested ourselves of them a few years back, and so unlike Roche, we don't have it internally. So we work quite collaboratively with diagnostics companies, and I think we still think that's the best way to work. So just to summarise I guess our progress within the company. The happy news about Iressa is that we did eventually do a prospective study with Iressa called IPASS, and it showed that in EGFR mutation positive non-small cell lung cancer patients it was superior to doublet chemotherapy, and so we launched it in Europe I think the year before last. It's still not launched in the US, which is an interesting thing. I'm assuming we're going to go for that next. Internally, we have a strategy for personalised healthcare, so that so it covers all aspects of the research and product development. So all our clinical development teams and our what we call discovery medicine teams, which is the early sort of crossover phase teams, have to review all the projects to see whether there should be a personalised element to it, if there are particular biomarkers that have been discovered that could be used, and have to report that when they put their at two different stages as the projects go through the pipeline. As I say, we're taking a collaborative approach to working on diagnostics and markers, and we've got a few collaborations and partnerships ongoing, the most recent one being a partnership with DACO that we announced quite recently. So to summarise, personalised healthcare is at an exciting time, and it's here already in terms of oncology, and I think it will have the potential to benefit everyone, because I think it will stretch over the whole of the healthcare portfolio. And it can work well, but it's also can be very challenging, and we need to assess the utility of it at as early a stage as possible in the drug pipeline. And again, I can't iterate strongly enough that you know, drug companies cannot do it on their own, and they have to work with healthcare agencies and other companies as well, and probably patient groups in the future as well, to get it working.